In this video, we're going to explain what DNA fingerprinting is and how it works. In the same way that the ridges on our fingertips or the patterns in our irises differ, there are regions in our DNA that are unique. While more than 99% of the human genome is identical across all individuals, polymorphic regions vary significantly and can therefore be used to establish a DNA profile that is unique to each person. DNA fingerprinting is commonly used in forensics, a field that applies science in a legal setting. For example, investigators often collect DNA in the form of blood, hair, semen, saliva, or tissue samples from crime scenes. These samples are then analyzed, and the resulting DNA profiles can be compared with the genetic fingerprints of suspects. Paternity investigations often rely on DNA fingerprinting as well. Here, the technique is used to compare the child's DNA with the alleged father's DNA. Additionally, in situations such as natural disasters, DNA fingerprinting can be applied to teeth or bones to help identify the victims. Beyond its use in forensics, DNA fingerprinting has found applications in multiple other sectors, including diagnostics, medical research, ecology, microbiology, and genealogy. The primary method for DNA fingerprinting is STR analysis, which stands for short tandem repeat. As the name suggests, instead of analyzing all polymorphic regions, it focuses on short tandem repeats, specific loci that contain sequences of 2 to 6 base pairs that are repeated multiple times. Since the number of repeats is highly variable among individuals, and since every individual has 2 alleles of each loci, one inherited from the mother and one from the father, they provide an ideal basis for DNA fingerprinting. Here's the step-by-step process of performing STR analysis. When a sample reaches the lab, the first step is to extract DNA from the cells. To ensure that there is sufficient DNA for downstream analysis, STR sequences are amplified. In this example, we're amplifying both alleles of 2 different loci, but in reality, you would analyze many more. The STR sequences are amplified using PCR, with a specific primer pair for each locus that attaches a unique fluorescent tag to the resulting products. After amplification, the DNA sample is run through a capillary gel electrophoresis system. Its capillary contains a gel that allows smaller STR sequences to travel faster through its pores. At the end of the capillary, a laser beam and a detector register the fluorescent tag of each sequence. The combination of the travel time and the color of the fluorescent signal allow the capillary gel electrophoresis system to determine the length of the STR sequences for each locus analyzed. If there are 2 different alleles, 2 fluorescent peaks will be detected. If the 2 alleles are identical, 1 fluorescent peak will be detected. The final step involves comparing the genetic fingerprint obtained with that of another sample. We hope that this video answered all your questions about DNA fingerprinting. If you're interested in more in-depth information on techniques other than STR analysis, please read the blog post linked in the video description. The blog also discusses method limitations and ethical concerns surrounding DNA fingerprinting. Thanks for watching. If you like this video, don't forget to subscribe to our channel for more.